Today we have the fine adventure of talking about ANOVA. If you're like me, the term ANOVA might leave you a little bit concerned. You've seen some of these amazing things people do with ANOVA to look at different effect sizes from different contributing factors in very complex studies, and you may think, nope, t-test is where I'm going to live. But I'm going to tell you that ANOVA, in its simplest form, the one we're discussing today, is actually quite handy for you. If you were trying to ascertain several different levels of a thing, not just two levels of a thing, ANOVA is going to come in very handy for you. So we've been thinking a lot about t-test and u-test, the difference tests of the last lecture. But I want to remind you that those are only useful for comparing one cohort to another cohort. What happens when you have more than two cohorts? So we'll talk about some examples along the way that, for example, would let us compare the measurement of a cytokine when there's no infection, when there's an H37RV infection, when there's an M. smegmatis infection, etc. These being different levels, where the infection type is the level. And you want to ask whether any of these infection types resulted in differences for a particular cytokine, for example. So we're going to try to walk through that. I'm shooting audio up here, Christian, so we're all set. We're going to walk through how we would use Oneway ANOVA, which is kind of another way of calling one-factor ANOVA, which is the simplest of applications in ANOVA, and I hope that that gets you at least a running jump when you want to move to something like two-way ANOVA, something that allows you to evaluate two factors at a time. I can give you an example of how a two-factor study would differ. Some friends of mine over at the University of the Western Cape are looking at different plants, in this case maize and sorghum, and they're evaluating them under wet conditions and under dry conditions. So we have in effect two factors, each with two levels. Is it maize or is it sorghum, and is it wet or is it dry? So that's a two-factor ANOVA. That's a more advanced application of ANOVA than the one way that we're talking about today. So let's try to walk our way through that, and along the way we're going to have some fun with our friend Tukey and the honestly significant difference test, and from there we will go on to have at least a little coda about the subject of multiple testing, something that I take quite religiously. So we're going to start with two giants, the first in the field of biostatistics and the second in the field of computer science, John W. Tukey and R.A. Fisher. I think that both of them are interesting to talk about. From there, we're going to move on to our test of are all of the cohorts the same, where you have one level. Maybe you're looking at plants under drought-like conditions, under a little bit of water, under a little bit more water, and a whole lot of water. You could treat those as like four levels of one thing. So under circumstances like that, are all of these cohorts the same? That's really the question that ANOVA attempts to address. And just as we've talked about the student's t-statistic and the student's t-distribution on Tuesday, we're going to look at the f-statistic that we use in the world of ANOVA. Then we're going to ask the follow-up question, because ANOVA will tell you something is different here, but it won't tell you what is different here. For that, we need Tukey's honestly significant difference test. So we'll talk about that. And finally, we will ask, why not just do everything with t-test, where you have a comparison between A and B, and a comparison between A and C, and a comparison between A and D, and then every other pair within that mix. So let's start with R.A. Fisher. I think this is a photograph of him that probably shows up in a lot of books. I'm going to come back to this photo in the next slide. So I want to mention he was born in 1890. He is towering in every sense of the word. The man is all over the history of biostatistics. He is largely credited with having created the field of experimental design, so it's a pretty high claim to significance. And he's also very important in making population genetics a real thing, being able to understand alleles in population, etc., working with Haldane and Wright. So these are all towering figures of modern biology. He wanted to serve in World War I, wanted being probably too strong a word for it, but he was chronically short-sighted. And so he was not allowed to fight, and as a result, we have the field of biostatistics. So I want to note also, though, that he was mercurial. Does everyone know the word mercurial? Mercury is reputed to be, I'm going to really botch the astrology here, but if Mercury is in your sign, you are thought to be a troublesome person. So he was a mercurial person and is notable for the long-running feuds that help propel some really great science. I mentioned that gossip, for example, is considered as sort of a tempering effect in the British statistical community. R.A. Fisher would be one of its lightning rods. All right, now he's born in what year? 1890. But I want to note that that photo came from the first International Eugenics Conference of 1912. So how old was he in that picture? 1890. He's 22 years old. So at 22 years old, he was one of the organizers at this first International Eugenics Conference. So let's talk about the skeleton in the closet. Biostatistics has a very lengthy history, a tightly connected relationship with the field of eugenics that led to such wonders as the Nazis in World War II. So I just thought you should be aware of this. Let's read this little quote from the citation at the bottom here. Fully 1/3 of Fisher's greatest work, the 5 concluding chapters in genetical theory, are devoted to an analysis of the decay of civilizations and its prevention by raising the fertility of more prosperous or superior social groups. Does that sound like anything to you? Have you heard anything similar from someone, say a sociologist who was once the leader of South Africa, for example? These ideas have strong relationships. And I don't want to just plow ahead and talk about R.A. Fisher as some sort of god, when in fact he has a very strong relationship with the eugenics community and is one of its leading lights of sorts. I want to note that it would be inappropriate to single out R.A. Fisher for having been in league with eugenicists. Not being in league with, in being a chief enabler of eugenicists. The person who built arguments for eugenics using biostatistics, right? There were lots of people in the biostatistical community who developed their theories as a way to support their work in eugenics. So you should be aware of this. On we go. He is one of the most notable and quotable people in biostatistics. We are not going to throw the baby out with the bathwater and say, because what he did was largely in service of eugenics, we must throw it all out. We're not going to do that. Because his theories are everywhere, and we need to be aware of them and understand them. So we're going to start with one of his best quotes that came from a statistical conference in India. To consult the statistician after an experiment is finished is often merely to ask him to conduct a post-mortem examination. He can perhaps say what the experiment died of. Talking to the statistician at the last hour is the wrong way to go. You want the statistician involved early in study design so that you have the power to achieve what you want to with that experiment. So he was one of the first to really point this out. What are the things that we look to R.A. Fisher for? Why is he such a towering person in this field? Well, he created ideas like maximum likelihood, an approach that is used all over the place. He was one of the folks to recognize that a sample mean is not the same thing as a population mean. What does that mean? I could ask, what is the average height for men in the world? And you might say, well, you'd say it in centimeters, and I wouldn't understand you. But I might say it's 5'9", for example. That's an American average, but let's say it's 5'9" for a moment. If that is the population mean, does that mean if I sample 100 people from a college campus that I'm going to get that same 5'9" represented? I'm not. That difference is to say that the sample mean is not the same thing as the population mean, an idea that R.A. Fisher put into publication. He was the person who defined variance. Would you say variance has an important role to play in biostatistics? Assuredly you would. We're not going to throw out variance because R.A. Fisher supported a lot of racist claptrap. Sorry, that's a rude way to put it. Mendel to Darwin, that may not sound like much of an accomplishment. Everyone understands that Mendel's theories about genes had something to do with Darwin's theories about evolution, about selection, and about variation, and so on. But in fact, this was actually a very difficult thing to accomplish in the early 1900s. People were finding it very difficult to explain why Mendel's results supported Darwin and vice versa. The idea that these stitch together in some sort of reasonable way. Some people call this the modern synthesis of molecular biology. R.A. Fisher was very important in that. ANOVA is R.A. Fisher's invention, so we must talk about him there. And I would point to this little book he wrote, The Statistical Methods for Research Workers, one of the first attempts to try to guide biologists in how statistics could help them. So, very important figure in this space. Let us now talk about a different figure. Tukey is a generation later, right? We were talking about 1890 birth year for R.A. Fisher. Here, Tukey was born, after that conference photo was taken. Tukey was a weird little Yoda figure in some respects. When he was a professor, one of his favorite things to do was to lie on his back with his eyes closed when the department was trying to figure out when and where can we schedule all of our class exams. Right? You've got these 15 classes, you've got these several spaces in which students can take their exams, and you've got this block of days. How do you set it up so that no student overlaps and has to be in 2 different classrooms at the same time to take the same test? That's hard, right? Tukey liked to lie on his back and just magically make these schedules in the air. He was kind of crazily known for that. Okay, so an interesting critter. Have you encountered some interesting critters in our department yet? I'm seeing a few nods. Some of you not yet, but it'll come, trust me. All right, so he was born in 1915. He was prodigious. The man was just obviously a genius from a very early stage. He was a very original researcher in statistics and had a tremendous impact on telecommunications and computer science. So we're going to talk about some of the things that he created along the way, and I think you're going to be very impressed with him too. He finished his PhD in just 2 years. Is anyone on track for that? Aiden, you going to get your PhD next year? No, it's going to be too long. Okay, that's fine. Well, he achieved the rank of full professor by age 35. Now, was that early or late? When did I make full professor? Does anyone know? I made full professor in coming here. I was an associate professor at Vanderbilt, so I made full coming here. And that would be 2015. I was 42 years old. That's pretty fast, actually, for a lot of people. So to get full professor at age 35 is kind of shocking, frankly. No one should make that your life goal. You're going to ruin your life if you do that. Okay, so what did he do? Well, we mentioned a little bit about bits and bytes. I think everybody remembers that from the start of the bioinformatics segment. The term bit came from John Tukey. The term software came from John Tukey. So definitely in at a very early stage, and one of the people who realized that certain concepts need terms laid to them. I enjoy this quote about him. The best thing about being a statistician is that you get to play in everyone's backyard. If you're a numbers person, if you're a numbers guy or a numbers gal, you get to work on everybody's project. Maybe you'll end up being middle author on everything that comes out of your department, just because everybody needs somebody who understands what to do with these numbers I've got. And I think his quote here really speaks very well to that. So ANOVA was pretty cool, but wouldn't it also be nice to have confidence intervals on ANOVA? And John Tukey was the person who put together ideas like that. Back when we spoke about sequencing, we talked about Fourier transform as a way to develop Phred scores, something for inferring frequencies in regular repeating data. FFT came from John Tukey. Box and whisker plots. We've all seen box plots, right? Right? We like box plots pretty well. Well, the rules that R uses by default in making box plots were those that were originally created by John Tukey for his initial things, as initial plots of that sort. He was a very big proponent of exploratory data analysis. Do we have a lecture in our biostatistics segment on just exploratory data analysis? We probably should. EDA is very valuable. It's the observation that before you go run into your statistical toolbox and do a bunch of statistical testing and so on, that you should look at the data, figure out how they distribute. Think about correlations among the data. These are this idea that the data deserve a look before you start applying tests to them is actually quite valuable. It's useful for quality control, assuredly. And robust statistical methods. We've already encountered one of these 2 days ago. We discussed U-tests and W-tests, the Mann-Whitney or Wilcoxon tests. So those are examples of robust difference tests, robust statistical methods. Okay, now I'm going to take a brief moment. Any questions about these two towering figures? I hope you don't mind if I spend a little time on history and biography, but I think it really helps to understand these figures a little bit better. Okay, now that I've engaged in some character assassination, let's talk about one-way ANOVA. ANOVA is short for Analysis of Variance. If I were writing a multiple choice question, I might throw something like that at you, but I'm not doing so. That's fine. Let's just move ahead. So, you measure an independent variable for samples that come from more, that should be dependent variable, I made a screw up. You measure a dependent variable for samples that come from more than 2 groups from the population. Does the dependent variable reflect group identity? So, if I were weighing dogs, for example, this dog is a husky, this dog is a mongrel, this dog is a poodle, this dog is a chihuahua, and I want to ask, does the dog breed impact the weight of the dog? We might find that some kinds of dogs have very similar weights. Maybe Labradors and Collies have similar weights. Anyone know on that score? Where would you go on Labrador versus Collie? Are they similar in weight? Don't know, okay. Peking geese, small, right? Chow chow, heavy, right? So you might have different levels of this factor, where that's the dog breed, and you have a bunch of weights, and you want to know which dog breeds are differentiable on the basis of weight. Imagine that you just have a dog at the pound and you're trying to figure out its breed. On a scale, you might be able to determine this is not a toy poodle. I don't know. All right, so we want to determine whether this dependent variable, the weight, reflects the group identity of that item. So if I'm weighing fruit, I might have an apple that has a certain weight, a pear that has a certain weight, bananas and oranges. I could use that for differentiating which of these fruits are differentiable solely on this basis. So why is it called Analysis of Variance? ANOVA compares variance under 2 different models. So the first is that we're going to compute this F metric, where we are asking how the explained variance compares to unexplained variance. So if you are ignoring dog breed and looking at the weights, you can compute the variance for that population of dogs. Then you can operate again by computing the variance, saying that these dogs are of a class. What's their variance? These dogs are of a different class. What's their variance? So this F statistic is a ratio of these variances. If those variances are very similar to each other, where between group and intergroup variance is the same essentially, then you don't have a basis for claiming that A's are different than B's are different than C's. So the F metric is our way of characterizing whether group membership is an important factor in determining this dependent variable. So the cool thing is that F is very similar to the t-squared metric. So students' t-test produces the t metric, the t statistic en route to a p-value. One of the things that people don't understand about ANOVA one-way is that if you run ANOVA one-way on 2 groups, you're effectively doing the same thing as a t-test does. So in some respects, it's easiest for you to think of one-way ANOVA as a multi-class, not just 2-class t-test. That makes it a little friendlier, I think, a little easier for people to understand. T-test only lets you do cohort A versus cohort B. ANOVA one-way is what you move to when you need to compare A's to B's to C's to D's. All right, so that's handy. Just as there's students' t-distribution for translating the t-statistic from a t-test into a p-value, the F distribution allows us to do the same for converting F statistics into p-values. And here I'm showing that some of these, in this case we're looking at different degrees of freedom shaping how these F statistics play out. Okay. So if ANOVA is wonderful and solves all the problems of the world, it does not, why do we need the Tukey honestly significant difference test? One-way ANOVA is willing to tell you that these different groups are not the same, or they are the same. Well, it's going to allow you in the same way as t-test to reject the hypothesis that they're all the same, the null hypothesis here. But ANOVA by itself will not tell you which groups are in difference. So imagine our case of the dog breeds. we had collies and Labradors, and we decided that they were similar in weight, so we couldn't really tell those dog breeds apart on weight. But the Pekingese, we could tell apart from both of those quite easily. Pekingese? How do you pronounce it here? Pekingese? All right, I'm hitting the G kind of hard. That might be an American thing. It's all right. So we have these different dog breeds. It might be that you can't tell a collie from a lab on the basis of weight, but you can tell a Pekingese. Is that what you want? A Pekingese versus a collie easily, and a Pekingese versus a lab easily. You see what I'm saying? So these relationships, knowing that they're not the same, isn't going to tell us which groups within those differ. Tukey HSD is for that purpose. Now, you might think that if ANOVA says the group means are different, you should go hunting after this with a bunch of t-tests. And we're going to talk about why you must not do that. The probability of falsely rejecting null hypothesis would be too high. The null hypothesis being these groups aren't different. They have the same means. So if you falsely reject the null hypothesis, you're claiming a difference exists when none may exist. That's a very bad error. We'll talk about that. So Tukey is going to find us a difference between classes without giving us bunches and bunches of false positives. So in this case, I've borrowed a study that somebody in the department was doing, and I've adapted it for my purposes. These are fake data. These are not a student's data. Trust me on this one. So we are going to simulate data for an infection study. I know this might seem really odd. I mean, we're always getting our tables from instruments, right? So why simulate data? I want to simulate data because I want to emulate the process of differences in data where I know what the differences really are. So let me try to walk you through this code. Can you see the screen okay? Okay, you have the PDF there, hopefully, so, okay, that's good news. So I'm going to set up for a study. Shall I stand right next to it? Would that be best for the camera? So we're going to have 5 replicates. Everyone knows you shouldn't do a 3-replicate study. Your PI will say we can only afford a 3-replicate study. You say back, well, we really need a 5-replicate figured out. You're the money person. Try that conversation sometime, see how it goes. All right, so we're going to have 5 replicates of each cohort. I'm going to use this model where we have 5 different infection states. So 1 factor in 5 levels. Does that term make sense for everyone? 1 factor in 5 levels. So the uninfected, the M-smegmatis infected, the BCG infected, the MTB attenuated infected, and the MTB H37RV infected. Do those terms ring a bell for anybody? Yeah, these are different mycobacterial cluster, tuberculosis cluster species. All right, now I've mocked up the data. I've created differences between some, but not between others. So what I have is, in effect, 3 levels. I have uninfected and M-smeg having a mean of 14. I've created BCG and MTB attenuated to give a mean value of 15. And then this last one, H37RV, gives a mean of 17. Everyone sees that? So we have 5 different levels, five different levels of the factor, but their measurements are only differentiated into 3 different groups. In effect, I haven't told you what we're measuring here because I don't even know. Maybe it's TNF alpha? Anyone good with that? Fine, so we're measuring TNF alpha, and you get the biggest bounce when you infect with H37RV. You get a little bounce from BCG and MTB attenuated, and you get no bounce at all from M-smeg because M-smeg is really not pathogenic in this circumstance. Okay, I'm just making this up. So we have these data that we're going to simulate, and in each case we're grabbing from the normal distribution 5 values drawn from a mean of 14. I didn't list what the standard deviation is. What's R's default for the standard deviation when you sample from a normal distribution? 1, exactly. Okay, so these all have the same standard deviation, but they have different means. Now, we're going to stitch that together. We're calling this whole thing cytokine, where we have these measurements as our cytokine levels, and then we're going to have labels that are 5 copies of uninfected, 5 levels of M-smeg, 5 copies of that, and so on. So I create 2 vectors, the cytokine readings and then the labels that apply to them. So everyone sees in their minds that we have 5 different replicates of 5 cohorts. Everyone sees that? So how long is my table? 25, exactly. So I have each infection state listed 5 times. And how many columns do I have?
5.
I have just 2 in this case. I have 1 column of the labels, of the cytokine numbers themselves, and another column of labels. So it's a 25-row table with 2 columns, the cytokine measurements and the labels. So we set out to have uninfected and MSMEG be the same cytokine levels. Does everyone remember that? Because both of them have the mean of 14. How did it pan out? I'm now doing a box plot, thank you John Tukey, where I'm plotting cytokine level as a function of labels. Did everyone see that? You use formulas a lot in R, and one of the ways you can use it is within a box plot. So here I'm plotting cytokine level as a function of the label using data from this object, this data frame, infection study. This is how I built it. So these are not just data I read in with read table, they're data I simulated in memory and produced. So what we see is that uninfected and MSMEG should both have means of 14. Do they? This is a reflection of something we already talked about, that the sample mean may be different than the population mean. The population mean, we know, should be 14 here. But we pulled out 5 numbers from those normal distributions, and the means we computed from them differ from what the true mean should be. Okay, so generally speaking, uninfected and MSMEG should be the lowest, right around 14. BCG and MTV attenuated should be averaging around 15. We see that BCG was a fair bit lower and the attenuated was a bit higher. And then the average for H37RV should be 17, should be up here. But its median value is actually down below it a fair bit. So this is an important observation. The data we observe are drawn from a distribution. They're samples drawn from a population. Samples, even when you have 5 of them, may be a little misleading as to what the means are. Everyone sees that? Okay, good. So these are some data I mocked up. What do we get when we run ANOVA on this and Tukey? So I'm going to do my ANOVA in 2 stages. I'm going to create an analysis of variance object in which the cytokine level is a function of the label. The data are drawn from infection study. This is exactly the code I ran, so you should be able to just enter this right off the slide and get exactly the same result. So I've created an analysis of variance object, and then I ran ANOVA on it. When I look at the result of ANOVA, I see I have 4 degrees of freedom. Where's that coming from? Very nice, yes. I have 5 levels of infection. Uninfected, MSMEG, blah, 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 blah. We have these 5. And degrees of freedom is frequently the number of different levels minus 1. So the degree of freedom number is 4. We have a sum of squares, a mean of squares, an F value, and a probability of getting a higher F value by random chance alone. That should ring all kinds of bells for you about the t-test. Because in the t-test, the p-value that results is the probability that you would get a more extreme t value by random chance. Here, the ANOVA test is giving us is evaluating the probability of getting a more extreme f value by random chance. And it puts this pretty little star on it. Now, if you actually run this in R, you will see that it defines what each of these little symbols mean. You can think of 3 stars as being wow, that's really significant. Sorry, it's a terrible thing to call it, but that's more or less what it means. It also computes residuals, which is to ask, in the model that is created, where we have some mean for each label, how much distance do we see within these classes? Within the MSMEGs, what was the variance? Within the uninfected, what was the variance, etc. So the residuals evaluate how well the data conform to the model that's inferred from them. Residuals essentially says how much is each point away from the modeled value for it. Okay, so when we run a Tukey honestly significant difference test, we get these back. Now this is probably a little confusing. We started with 5 values, and suddenly we're getting this big roster of paired things. I talk a whole lot about the handshake problem. How many people you have at a party versus how many distinct handshakes you can get. This falls right out of that playbook. We have 5 different levels. So how many different handshakes do we have among 5 people? The answer is 5 times 4 divided by 1. Divided by 2, my goodness. So that's 10. So you can pair up uninfected with M.Sm. You can pair up uninfected with BCG. You can pair up uninfected with the attenuated, or you could compare up uninfected with H37RV. Everyone sees all of those comparisons involving uninfected. You can go through that. So in effect, Tukey HSD is going to try to give us a significance level on each of the pairwise comparisons within this set of 5 things. So the result is M.Sm versus BCG, MTB attenuated versus BCG, H37RV versus BCG. So we've got this table back. Now for the moment, I just want you to pay attention to this last column. These are adjusted p-values. And you probably saw adjusted p-values once before when we were talking about microarray data. This is an example of multiple testing correction. Tukey HSD is going to compare these individual pairs within these 5 levels and give us a corrected p-value that reflects whether or not these are differential or not. Okay, so our ANOVA told us, yes, there are differences between means somewhere in this data set. Tukey HSD tells us where they are. So how do we read this? This first pairing, is that a significant p-value? No, that one stinks. That's 0.9. That's a terrible p-value. Nothing. What about this one? You like that? That's a p-value of 0.014. That's a good. So we're going to put a little star on that in our minds. 0.53 lousy. 0.34 lousy. 0.002. Okay, that's one we can put a little star on. 0.94. Not any good. Nope. 0.129. Pretty hinky. 0.099. Anyone in favor of keeping 0.099? No one's in favor. That's good. We throw that one out too. And finally we get down to the last one, 0.0004207. We've got a thumbs up on that one. Okay. Now, the value of using a simulated data set is that you know where the differences really are. Remember, there were 3 levels of these things. So we should be able to tell uninfected and M.Sm apart from something that's similar, but not very powerful, the attenuated result and the BCG result. They were slightly different. So let's try looking at those. Uninfected BCG, these should have different results, correct? I think I said BCG had a little effect. It's the mid-level. But we see that the p-value for it is not significant. So that's already one hammer down. The variance was great enough that we weren't able to tell those 2 apart, even though we know that they were sampled from distributions with different means. Does everyone see that? By using simulated data, we're able to check whether this statistical model is capable of what we're trying to achieve.
So, sir,
yes?
If we had increased the sample number, would
yes?
we manage to see the difference?
You might. In fact, if we just ran this again, because each sampling of this is random, it could be that if I simply ran it again, it would be an obvious, glaring difference. But in this case, the data that we sampled for BCG and uninfected were not so different that they were differentiable by ANOVA, by the Tukey HSD.
So that sample is messed up?
The sample is drawn from the correct population, we know. It's just that it's a random outcome. You don't know whether if the uninfecteds are read a little bit high and the BCGs are read a little low, just by random chance, You can't differentiate them. So it's useful in this case to simulate the data to ask, how big an effect size do you need before you can detect something? Effect size is something Guillen really wants you to understand. So I'm trying to be a little, to punch that term when it shows up. In this case, we ask, how different are the means, but while we take into account the variance that we have in these populations. If you've got a big variance with a small mean difference, you can't detect the means being different. If you've got the big mean difference and small variance, you're in a great place. So in this case, the variance was probably a little too high in this particular sampling for us to recognize that difference. So what are the common factors for the significant p-values that we got here? H37RV versus BCG is one of them. H37RV versus M-SMIG was another one of them. And H37RV versus uninfected. So what we had in common for all of these significant p-values was that the most extreme categories of differences were sought, with the exception of H37RV versus BCG. Those were the BCG was our middle level, and we saw that in this case the extreme values of H37RV were differentiable from that. Okay, so again, you should realize that statistical tests may fail to pick up differences that are real. If we crank this up to 10 replicates, do we have a better shot of finding differences? Assuredly. Is your PI going to afford a 10 replicate experiment? Maybe not. Maybe not. All right. Now, what happens if we do come in? Oh yes, Christian. Yes.
So when you have a large p-value, you can call it significant. You can say.
Right. Generally speaking, people use, and I'm going to kind of ignore a raging controversy in the field of biostatistics right now, and say the rule that people follow is that if you get a p-value, a corrected p-value of less than 0.05, you get to put a little star on it in your paper and say it's significant. This is how things work. The reality is that p-values get misused in a huge number of ways, and you frequently find yourself reading papers in which a student says, my p-value is 0.06, so this is tending towards significance. And statisticians in the audience all want to slap them immediately because you set the rule of 0.05, you live and die by the rule of 0.05. You don't allow yourself to talk yourself into keeping things that are close to 0.05. But there's a very strong disagreement going on in the biostatistical community right now about how one should set rules about what threshold is appropriate. I'm sure that Gerard will be more than happy to talk about that debate in some detail. Okay, good question. So now we're going to, oh yes?
Can I just ask with the 0.05 rule?
Yes.
If you did the, you ran the ANOVA once and then let's say it was above 0.05 and then you ran it again and then it was below, would you use?
I wanted to clarify that the reason why the p-values would change was because the data I fed it changed. In this case, I'm randomly sampling the data set that I'm simulating for this experiment. If I simulate the data once and run ANOVA 5 times, I'm going to get exactly the same p-value every time. The reason why it would change if I repeat the experiment, if I simulate a new batch of data, is that the samples would change. But if the data you're starting with haven't changed, you have no reason to think the p-values will change. Good question. Very good question. Okay. Great. Now, let's talk about what happens if you haven't taken a biostatistics module in your honors class. You might think, well, I know how to use t-test, so I'm just going to use t-test. You have had the opportunity of learning about ANOVA and Tukey honestly significant difference test, which means that when you present a master's proposal, or you even present the results from your honors project, if you follow this approach, one of your teachers will likely hold up the dreaded hand at the end of the presentation to say, why didn't you do an ANOVA? You have more than 2 classes. So what happens to people if they just run in what my computer science teacher would have called a brute force and ignorance approach? If all you know is the t-test, and you don't know how to use ANOVA, and you just careen in here with t-test, what happens? So I might run t-test on the uninfected set versus the mSMEG set, and then grab out the p-value. Everyone knows what this $ business is? We're grabbing out the p-value element from the list that is returned by t-test. So I might compare this set, I get nothing, nothing, nothing. Okay, so I have hits. This hit is for H37 versus uninfected. We feel pretty good about that. This hit is for H37RV versus mSMEG. I think that one is repeating the other result. And we have H37RV versus BCG, again a hit. In this case, the number of hits doesn't change. We had 3 hits in both, didn't we? But you'll note that the p-values reported here are different than the p-values we had before. That's because this t-test application doesn't know that you're running t-test again and again and again. So Tukey is correcting for the fact that you're comparing 10 different pairings. T-test has no information to that effect, and it cannot correct the p-values for you. So the p-values that come out of t-test are not reliable, whereas the ones that came out of Tukey are. So don't walk yourself into a multiple testing problem. Tukey honestly significant difference test protects against type 1 errors. This is claiming something's different when it's not. Rejecting the null hypothesis inappropriately. Tukey is going to protect you against that. Next, Tukey also protects you against beta errors, type 2 errors, which is to say claiming that there's no difference when there really is. This is another reason that those p-values are different. So Tukey is better on both sides of this equation. As I've mentioned, the number of tests you must perform increases with roughly with the square of the number of different levels you've got. In this case we had 5, therefore we had 10 pairings that we had to evaluate. How many pairings would you have to test if you had 6 levels? You plug in your 12, we had 5, we're going to change that to 6 levels now. So we plug in 6 in this equation for n. 6 times 5 divided by 2, 15. All right, so how many comparisons did we do with 5 levels? 10. Adding just one more level added a 50% increase in the number of pairings we had to compare. So Tukey HSD can do a lot of work for you and protect you from doing t-test after t-test after t-test. ANOVA with Tukey HSD is a natural combination. I know that I say this a fair bit, but just running a whole bunch of t-tests because that's the test you know how to use will frequently get you in trouble. So try to work out the best strategies for analyzing your data. In its simplest form, ANOVA is helping us to ask whether any differences are present among any of the levels in our data set. If you get a p-value above 0.05 from the F-test, from the ANOVA, you don't have to do the Tukey HSD. You already know there aren't differences to go hunting for in this data set. So there's a dependency here. You do your ANOVA. If ANOVA says there's no difference to find, don't go looking for it. T-test might find a difference for you, but it's not a real one, because ANOVA has already failed to give you a significant p-value. Okay, so Tukey HSD is a follow-on and is going to help you to find the differences when ANOVA has established that one is present, one or more. Okay, any questions about ANOVA and Tukey honestly significant difference test? It's kind of a cumbersome name, isn't it? That's why people call it Tukey HSD. Tukey HSD is not the only way that we have for evaluating ANOVA results, but it happens to be a pretty easy one to implement. So I recommend it. Any more questions? All right, so I think what we're going to do here is take a brief moment to reassemble them. We're going to turn off the recorders and get ready for the practical, which follows right on the heels of this. So if people need to take a quick 5-minute run somewhere, We'll be here and start up at about 10:30 on the spot. Is everyone good with that? Great, thank you.